Hi, everyone. We're here with Janani, who's a first-year graduate student, and she recently completed a laboratory rotation in the Miller-Jensen lab, where she used systems biology approaches to study melanoma, which is can be a very deadly form of skin cancer, especially if not caught early. So Janani, can you tell us a bit about what you were trying to figure out?
Yeah, thank you so much for that wonderful introduction, Doro. So melanoma is one of the most deadly forms of skin cancer. And like most cancers, it's really difficult to treat because of its ability to hide from our immune response, which normally finds and destroys abnormal cancer cells. And the most popular treatment option for this is a therapy that targets this immune response, called checkpoint inhibitor therapy, which removes the cancer cell's ability to hide from the immune cells, and therefore our normal immune response against tumors can effectively remove the cancer. However, this option is only effective for 50% of patients, and we want to understand why this treatment is not effective at removing the cancer for some of these patients.
Wow, that'd be great to make that an effective therapy for 100% of the patients. So how does systems biology actually help you figure out and ensure that the immune system is able to find and destroy cancer cells?
Yeah, so one way we can approach this question is to investigate the sources of such complexity in the immune system and tumors. And the immune response to tumors rely on a web of cell-to-cell communication pathways. And one example of cell-to-cell communication is an immune cell that recognizes and destroys an abnormal cancer cell. Now, in reality, in the tumor, there are actually several immune cells, tumor cells, and supporting stromal cells that are all involved in the immune response against tumors. And each of these cells has several different ways of communicating with other cells, and this forms the complex web of communication. So as scientists, we can use multiple technology platforms to learn more about how these cells are communicating and determine important interactions in the immune response that we can target, with the hope that what we learn about melanoma can be applicable to other types of cancer as well.
So how do you go about targeting one of these cell-to-cell communication pathways, or even know which pathway to target, given that it's this whole web of cells that make up these communication pathways?
Yeah, that's a great question. So we could start by using mouse models of melanoma to look at all of the cells involved in this complex tumor immune response without biasing which cells or which communication pathways we're looking for. And then we can use computational tools to evaluate these different cell communication networks that may be present in the tumor immune response, and then generate hypotheses about which pathways we can maybe target. And so this is the stage that we are in, trying to figure out the pathways that we can target. And once we have determined this, we can then test these hypotheses using simple cell models with just a couple of immune cells and tumor cells, and then expand this to more complex mouse models to verify these hypotheses. And together, the goal will be to establish different ways in which we can target and enhance immunotherapies such as checkpoint inhibitor therapy.
Wow. It's not the first research project that we have heard about where you guys are employing both wet lab experimental approaches and computational approaches to really get at your question that you're asking. Is this something that you enjoyed in this particular research experience?
Yeah, for sure. This is an approach to science that I really liked. And one thing in particular was combining computational and experimental approaches to solve a problem. Like, for example, as we get more data about the biology of cancer, we can use computational analyses to then glean important information about the patterns in these data. These hypotheses can then be incredibly informative for guiding experiments that may be most promising for solving our initial questions. And my hope in my PhD is that by combining the strengths of computational and wet lab experimental approaches in biology, we can understand the complexity of biology and then also propose simple solutions for diseases such as cancer.
Well, it will be exciting to see which lab you end up joining and what project you end up working on your PhD. Thanks so much for telling us about your lab rotation.
Thank you so much, Doro. I really enjoyed it.